as the answer. Okay, number one, some of you were missing out on the title. Normally in biology, the moment you are told to make a drawing, just know that you must always put a title. You must always put a title. If you don't put a title, you are missing out on the marks. So never miss a title. Number two, when we are making drawings, biology drawings, I want you to take note of it from today. A good biology drawing has particular features it must follow. Number one, we don't shade. Let me even highlight them. No shading. No shading like that. You must never and never ever try to shade. Maybe what do I mean by shading? I'm assuming you are drawing this upper part of the flower, and then you begin doing like this, like you're doing art. Okay, and then it's like you are doing art, and then I'm assuming you've done that. Then you do that, you do that, you do that, you do that. But the work is not clean. We do not entertain anything like a shade. It must be a smooth outline. As you can see here, okay, you can see in this drawing, everything is neat. Actually, we give a mark for neatness. We give a mark for neatness. Now, some of your drawings were not neat. You are using very thick pencils. The pencil must have a very thin outline. That you don't mix things like this. Okay, so that is what made some of you to have poor drawings. From what I saw, you should never have anything that is not clear. The outlines must be separate from one another. Okay. Number three, we don't entertain broken outlines. For example, I'm assuming you're drawing a leaf, and then it comes like this, and then it comes like this. You leave some space there, like that, Okay. Lucy is saying I didn't mark you. I've told you that there are some of you who have not yet marked due to some technicalities, but very soon each one of you will receive your paper when it is marked. And by the way, what I saw, some of you don't know how to scan. You are bringing diagram picture which cannot be read very well. Okay, and I remember they told you that you are supposed to scan one document and all the, but some of you were scanning number by number. Okay, so you open a document and it's like as if you submitted one, opening it and you find that it is only one number. So the next time you're handing in, please put all the numbers in the same document. Don't scan number by number. That was bad. Okay, so what do I mean by broken outline? So if I can use a blue pen here, this thing here, you see the line is broken. Even here, the line is broken. In biology, we don't want broken outlines in a diagram. For example, roots, some of you are drawing like the roots are like this. I draw like this, and then you leave them when they are open. That's a root, that's a root, like that, that's a root, like that. Leaving them like that. Now, these are broken outlines. We don't entertain them in biology drawings. So this one can make you lose all the marks on a biology drawing. Okay, for any biology drawing, always close all gaps. Are you seeing any open outline on this drawing? Okay, on this plant? No, all gaps have been closed. So that's what we do for biology drawings. Okay, so you must have a title, it must be neat, okay, and a magnification. But for the case of this, because you are just getting it from your head, even if you didn't include the magnification, but in case you are given a specimen to observe, whether in a microscope or just using a hand lens or using your eyes, you must always include a magnification, and it is always put on the lower right corner here. Lower right corner, you can come and put here X, let's say X5, meaning the magnification is times 5. Okay, so for some of you forgot this, and also when we are labeling, in labeling we don't cross lines. For example, you can do like this. This is a leaf, this is a leaf, and then you come and put here, okay, that this is a seed. This is not allowed in biology. When label lines cross, it's not allowed, and never do it again in as far as biology is concerned. As well as also scattering label lines. Some of you just like this. Now that's a leaf. Now this is let's say a vein of the leaf. This is a root. You see, you scatter them. For a good biology diagram, the lines must be horizontal. Like you can see here, all the label lines should always be horizontal. Okay. And what if, maybe you may ask me that, what if what you want to draw, okay, let me label. Let me show you what I mean. Okay, where are you? The pen is here. So I'm assuming you want to, let's say, label this flower, and this side you didn't have space. On the left side, no space. So you do like, and the only way, you don't want even to cross with this line. So what you do, you get a label line using a ruler, and then do like that, and then afterwards you make it horizontal like that. So it comes, then you make it horizontal, and then you label that this is the flower. This is a flower. And also, it's always advisable to label in singular. This is one seed, so you write seed. This is one root, so you write root. Sorry for that, for this one here. Emma has a question. Yes, Emma. Emma, I've asked, you've raised your hand and I've asked you to unmute. Can you please ask your question?
I was asking. What if you're doing the full flower?
Come again?
What's the scientific name for a male part of a flower?
Okay, thank you for that question. Emma is asking for the scientific name, the male part of the flower. Okay, now that one is normally taught under flowers, and we shall have to look at it a little bit later, but maybe to tell you what it is if you don't want to call it stamen, so you can either call it stamen. The male part of the flower is called stamen. If you don't want to call it stamen, you can call it androecium. 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 So you can either call it androecium or you can call it stamen. Okay, that's the male part of the flower. But of course, not all, some flowers have both the male and the female. I'll talk about it when we have reached the flower. Because so far we began with the root, then we did the stem. After the stem, we shall do the leaves, and then after the leaves, we shall do the flowers, and then fruits and seeds. That's how this subtopic flows. Okay. Then, okay, question there. Then I hope you guys have been able to understand the good qualities of a good biology drawing. So for the next time, you don't just draw anything for the sake. As a bio, you are supposed to be very neat as far as drawings are concerned. And they carry marks even when we are marking them. Although at this stage we are not yet strict. At this stage it's about to have you labeled. But when you reach senior 3 there, and we begin serious practicals, we begin always marking the title. Is always a mark for the title, for the drawing, for the labeling, for a very nice outline. We give marks for neatness. We give marks for magnification. You see, so it's not about drawing and you label for you to qualify for all the marks, but you must have all these other qualities of drawing, outline, no broken outline, neatness, and then magnification. These ones must always be there. Ubo is saying, did you mark our work? I'm still marking, and I marked, of course, some of you, but during the marking, the system got a problem that we are using to mark, and we couldn't do it, but be sure very soon each one of you will receive a script which is marked. Okay, so we can go to the next number. Number 2 is saying that roots are very important to plants. State any 3 importances of roots to a plant. 1, they anchor the plant family in the soil. Okay, that's one importance. So roots anchor the plant family in the soil. Okay, and then Tendo, you have a question? Yes, Tendo Brenda.
They give support to the plant.
Thank you very much. Yes, they give support to the plant. They give the support to the plant. That's also correct. They support the plant where, or is be specific. Supporting how?
Position.
In one position or in the soil?
Yes.
Okay. Okay, thank you for that. Then they also absorb water and mineral salts. There are some people who are putting up their hands. Irene, Chezowa, Chosaba, Irene. Okay, yes, Irene.
Roots conduct the absorbed water and mineral salts up to the stems and leaves for the plant.
Can you repeat? Can you repeat again? Irene. Where is she going? There's something which I wanted to hear from her. Irene, I think, has gone. So, Saba Irene, what do they do? There is that word which we have used. You see, there is this word which is absorbed, observed. These are 2 different words. Or adsorb is also adsorb. Okay, so those are you must be clear with which kind of word I didn't get the word which you used. Yes, Irene, can you unmute and you say the word which you meant?
I repeat the whole.
Yes, Irene, which word did you? No, just that word. Which word did you use? Absorb, observe.
I said they conduct the observed water and mineral salts up to the stems and leaves.
Observed.
Yeah.
The word is not supposed to be observed, Irene. When you say observed, what observed means you see. It means to see, okay? So the word is not supposed to be, when you use this word observed, it kills the whole point, okay? So the word is absorbed. Some people also confuse it with adsorb. To adsorb means to get stuck, it sticks onto the surface. But this is absorb, like it is taken into, okay? So you must use a proper word there. And then reste, yes, reste.
I just want to ask.
Yes, please ask reste.
The meaning of anchor.
Anchor, thank you very much for that question. Anchor. If you have watched movies, normally movies whereby there is a ship, normally there is a ship which is, let's say, floating on water. I'm assuming there is a ship floating on water. Let me get a blue, and this is water. Now, sometimes when a ship is water, and let's say it wants to first stop, you see when you're on driving, you can just stop the engine and then you park. But remember, this water is always moving. Water, if you've observed an ocean or a lake, water has waves that keep on moving the ship. So this ship can easily be pushed by the water to a distance which you even don't know. So, but what happens is that sometimes when this ship wants to first make a stopover, let's say in a given point on the ocean or on the lake, it has that anchor. Looks like it's normally a heavy kind of metal, very heavy, with a rope tied on it. So it is thrown and then it goes to the bottom of the ocean or of the lake. So the moment it sticks there, so this rope is going to be holding, it is held by the anchor as the ship is stopping by. So to anchor means to hold firmly. It means to hold, to anchor. It's like in Uganda, okunyweza. Okay? To anchor, to hold it in a given place. So to anchor just means to hold firmly. And then, okay, also they conduct the absorbed water and mineral salts up the stem. To conduct is like to push, to move, okay, to let flow, all that. Then we can go to the next. It says that write down any 4 examples of modified roots, modified root types. Examples of modified root types. Now, we had looked at different examples of modified roots. For some of you have forgotten. If I can just play this very, very fast, just to remind you of these modified. I think some of you had joined us before we had looked at this modified slideshow from current. Okay, remember we looked at modified roots. Modified roots, and we began with the storage roots, which included the cassava, For some of you had forgotten, it included the sweet potato. Sweet potato is monde, and then cassava. Cassava is a muwogo. You can see these are all modified roots. They are modified to store food. So what is that type of modification called? It is they are called storage roots. Storage roots, and the most common example is carrots, cassava, and then sweet potatoes. Okay. And that's why I got these pictures of a whole plant, Otherwise, many people may not understand it when they just look at it from just there. Then we also looked at breathing roots. This was also some other kind of modification, because primarily roots are known to, one, conduct water, cut the plant in soil, as well as absorbing water and mineral salts. So if it is not doing those primary functions, then what is it doing? So some roots can be modified to do different work apart from the other work which they are known for. So some can be modified that instead of absorbing water and mineral salts, they can have some roots storing food. Then they can also have some roots being used for breathing, especially those plants which are in waterlogged areas like swamps. And you can see this mangrove. If you want to see such kind of tree, I think I've seen this tree in, there's a place along Entebbe Road called the Zika Forest. At the end of the forest, there is some kind of a swamp, and some of these trees are there. If you entered that forest called Zika Forest, like the other end, most people, when they go to it, they just stop just midway, just to have to look at different types of trees. But when you go extreme, it is a part of a swamp which has some of these plants. And then some of them can be modified as stilt roots. Like here, they also just give you additional support because it has some roots already, but these ones are modified to give extra support. So some of you had forgotten that we had actually looked at this. Also, the prop roots, and remember we looked at the one for sorghum. For some of you have never seen sorghum, this is maize. These are prop roots of the maize, and these are prop roots of the sugarcane. The prop roots of the sugarcane plant. Again, they also give extra support. Then the buttress roots, we also looked at them as a type of modified roots. And these ones are found in very huge trees with huge trunks. And I think you've seen some of these trees, roots which are on top of the soil. They arise like that just to give extra support to these trees, because they have huge trunks. If they're only having the other small roots down, which absorb water, when wind comes, it would easily blow them down. Okay, but because of these strong and stronger buttress roots, you can't just blow them down. Then there are also clasping roots. These are some modified roots. So clasping, to clasp means like to tie, not to kusiba. So these ones they clasp, that is, they tie the plant on another support. Okay. This tree on the background, like this is a vanilla plant. This one here, these are vanilla leaves. For some of you have taken vanilla tea leaves, or you've taken yogurt containing vanilla flavor. They get that flavor from this plant called vanilla. It has a weak stem, you can see it here, but then it can grow roots and then it supports it. So normally people who grow vanilla, they actually like using sometimes they put their small bovule trees to support the vanilla plants, because they need to keep on growing up to trap sunlight energy. This was from this picture was from a plant. Is a plant called elephant ear plant. Elephant ear plant. It's the one which is like this. So very many people use them as flowers in their homes. Elephant ear, it normally has those huge leaves which look like the ears of an elephant. ears of an elephant, like that. So it's called elephant ear plant. Semare is saying too speedy. Now, Semare, we are we studied these things. It would be like too much repetition. That's why I just wanted to show you some of these plants because we had studied them. Maybe you have just joined Semare. Kasuti Nolin, you have a question?
Excuse me, teacher. Why are they?
Yeah.
Why are they particularly called modified roots? Like, do they have something which makes them modified? Like, I don't understand.
Okay, before you mute yourself, do you know the meaning of the word modified?
No, but I can't explain it.
Come again?
Okay, like the word, like modified.
Modify, what does it mean?
I can't. Okay, I don't know how to explain it. I really, I don't know.
Okay. Okay, let me explain to you the meaning. It will become a little bit easy. Okay. Tendo Brenda, you have your hand up. You have also a question. Tendo Brenda, you have a question. Someone called Dr. Nabatukuvu is saying, when did you start the topic of roots? We have been running this program for the past month. For some of you have just joined. I think most questions are coming from people who have just joined. When we are learning this, we began with the roots, okay? So we have been running this thing for some good time. Yes, Victoria.
Teacher, I was asking, like, if they ask for examples of modified root types, should we write the examples of the roots?
They wanted modified roots, so you pick out one, because even those modified roots have examples of plants that have them. You get it? For example, when I talk of, for example, modified roots, there are some that are modified for storage. Okay, so that's a huge group, but so the ones modified for storage, they are examples of plants that have them. For example, the carrot or the cassava, as I showed you here. Cassava, for example, when I talk of, let me first go to this picture of cassava. Cassava is when I talk of cassava, cassava is a whole plant, the cassava plant, okay? Which plant has leaves, it has a stem, and it has roots. So the modified root, the modified form is the storage roots, and it is possessed by plant. Okay, so when I give that, I hope you are getting it. Faith is saying, sir, we are not understanding you because you are fast. Oh, and yeah. Am I fast? But okay, what I was doing, what I was showing you is that maybe some of you have just joined. We had studied this long time, okay? So when I move slow on this subtopic, it's like it's like I'm reteaching what I had taught already. Okay, it's like I'm reteaching. It's like I'm repeating what I had taught already, because when we are beginning this topic, it's actually subtopic under kingdom plant. Okay, when you studied the 5 kingdom system, the 5 kingdom system, there are 5: kingdom animalia, kingdom plante, kingdom fungi, kingdom monera, and kingdom protoctista. Now each kingdom has phyla, so we studied different phyla in each kingdom. Now under kingdom plante, there is a phylum that called angiospermatophyta. Okay, Phylum Angiospermatophyta, also known as the flowering plants. Now, under the flowering plants, this is where we study a whole plant from the root to the stem to the leaves, because we have not yet even done leaves. So after stem, our next topic is supposed to be leaves. Then from the leaves, we also study the flowers that are produced by these angiosperms. And then after studying flowers, we also study the fruits produced by the flowers. From the fruits, we also study the seeds which are in the fruits. After that, then we can look at dispersal mechanism. Okay, so you must be able to know. Sir, you repeat, I have just joined. Now I tell you saying you repeat, I have just joined. That would some people who already joined, it will be a disservice for me to repeat the topic which I had taught already. Okay, and we teach according to scheme of work, so there is more work to cover. So what you have to make sure is that don't miss lessons and always come in time. For example, some of you have just been and 30 minutes so far passed. Okay. Okay, so let us I've just shown you the prop roots, then buttress roots I've shown you, the clasping roots I've shown you. We also have those epiphytic roots from epiphytes. Epi, it's a Greek terminology which just means on top of something, okay? And then phytes just means plant. It has to do with plant. So epi on top, phytes on top of plant. Okay, that's why when you're looking at angiospermatophyta, angio, that's flowering, then plants. Okay, gymnospermatophyta, non-flowering plants. So this phyta word, in any word that you find containing phyte, just know it has to do with the plant. Now, this one, for example, in Uganda, there's a plant called Enzilugaze. So normally you find that there is a tree, and on top of a tree, there is a plant growing on it, which is not part of that tree. So those are called epiphytes. So the roots that support them, that enable them absorb moisture, so that they can get hydrated, those are called epiphytic roots. So I think we can go back to our question. We can go back to our, where are you? Corrections here. So let me play from the current. So we had looked at, so what you are supposed to do here, these are the answers of modified root types. Then we can go to draw a well-labeled diagram showing the transverse section through a bean root. Okay, now this question was somehow technical. And how was it? A bean root, much as you've never seen a bean root, or I could even have used, let's say, a mango root. So whether they use mango root, bean root, or fene is called jackfruit root, as long as that plant you've detected that it is a dicotyledonous plant, okay? As long as you have detected that it is a dicotyledonous plant, then the transverse section remains always has the same pattern and same drawing. If they use, and how do you know that it is actually a dicotyledonous plant? Someone is saying that, can I get the notes from Google Classroom because I have missed a lot, but I have been attending? Yes, the notes are there already uploaded on Google Classroom, and you can always get them. This is very slide, it's there. So how do you know a dicot? Let me write here a dicot to represent dicotyledonous plant, and then on this side let me put monocot. Monocot. Of course, with biology you write the whole word. Now, if you want to understand what a dicot is, think about any plant that looks like a bean plant. Okay, think about any plant that looks like a bean plant. What do I mean by looking like a bean plant? When you examine a bean plant, it will have relatively broad leaves. So a plant with broad leaves should fall under a dicot. It will have network of veins. You see, a plant normally has veins in it, veins in its leaves. Where are you? I'm looking for the cassettes here. So normally you find that these leaves will have network veins. They just run like that, like that, like that. So those kind of network veins, sometimes they are called reticulate veins. We shall study more about this under the leaves. So when you see any plant that has network veins in its leaves, just know that that plant is a dicot. Okay. Then how does a monocot leaf look like? Now, any plant that looks like grass, I mean grass is obusubi in Uganda. In Uganda, grass is called obusubi. Even some other languages, I think it doesn't change much. I don't know much of the other languages. So, but just know grass. Normally, grass will have a narrow leaf, lamina, or blade, and it will also have parallel leaf veins. Their veins never meet anywhere. They are just parallel. They never meet. They're just parallel. First of all, the leaves are narrow and they are parallel. So that is a monocot. Okay. So I think when I was teaching roots, I showed people how we actually end up with this structure here. Sorry for that. So I taught people of actually how we end up with this. And I had even showed them a picture, which is which looks a little bit complicated, but of course it's not, because that's how actually it looks like when you observe it in the microscope. Because in a microscope, this is not what you see. Okay, we get a thin section of the root transversely, and then we place in a microscope and we begin observing. So this is not what we see. What do we see then? For some of you forgotten, let me show you what I showed these people. Okay. Okay, let me run here. Here. So actually, let me first make a slideshow here from current. Yes. So in a microscope, although this one is colored, if you want it colored, we normally stain. We stain using certain stains in the lab. But at this level, you don't need to know the types of stains which we use. So after staining, it's when you can see such a structure in a microscope. But after seeing this is not what we draw, okay? This is what you see in a microscope. We extract what we call a tissue plan, and this is what we draw. Just layers, so you can see that this is the outer layer of the endodermis, or you can call it, sorry, of the epidermis, or you can call it the peripherous layer. So it is having 2 layers like that. You can see it there. It is having 2 layers and root hairs on it. Of course, here they showed only a few, because root hairs are very easy to damage when you are cutting off a thin slice. Sometimes you may never find any when all of them have been damaged. And then this one is drawn using 2 lines. You can either call it epidermis or peripherous layer, like that. Dermis just means skin. That's why a doctor who studies skin, there are some doctors who specialize in skin. They're called dermatologists. So they are specialists in skin. Okay. After that, now you're seeing these small blocks here. These are cells. Now, there is no way how we draw these cells, so we just leave space here. So we don't put anything in the middle for these cells. Then we draw another layer. It is just drawing using a single line, and then another inner layer, which is the pericycle. You can see that is the other layer. Now here, observe it. You can see that the xylem is actually in form of a star shape. That is how the xylem is, but of course also made up of cells. You can see. So, but we don't draw the cells inside, we just draw this star shape, okay? And then on the side, you can see those structures there. That's the phloem. That's the phloem, and that's the phloem. So the phloem is actually located on the side. You place this phloem on the side. So this is what we are supposed to draw here. But unfortunately, some of you drew this one for the monocots, for grasses. This one is saying, help me with the timetable. The admins already, and even today, I saw it being shared. I don't know, maybe you are not on WhatsApp, I don't know. Maybe some of you are not on WhatsApp, but everything is always communicated on the timetable about any progress, unless you are not on the WhatsApp. So, so this is what you are supposed to draw. And as I told you, when we are marking, we follow the other qualities of a good drawing, okay? The title must always be there. Neatness, no broken outline, okay? We don't shade. Now, for example, here, of course, here in this picture, this ball, this phloem was shaded, but in the right sense, it's just supposed to, let me draw it here. That's the phloem. The xylem, you just draw some circles and you don't shade them like that. We don't shade in biology drawings. We don't shade. Can I get the WhatsApp number? The WhatsApp group, I think it is even already full. I may join in the rest of my friends. The WhatsApp group, I'll check, and then I shall share the link, but I doubt it's most of the WhatsApp groups are already full. Someone had raised their hand. I'm not seeing them again. I think they had a small question. So for some of you who drew this was for the grasses, and this is how it normally looks like for grass. But grass, actually, let me give you examples. Grass, there is maize, casoli. Actually, most of the cereals, rice, all those ones. Green, you have a question. When I ask you to unmute, you unmute and you ask. Yes, Green. Good afternoon to you. Yes, good afternoon. I can hear. Please ask.
I just wanted to confirm whether I'm on the WhatsApp group, because I'm not seeing the WhatsApp group on my WhatsApp, and I wanted to join.
I think you are not there if you are not seeing it.
Then how can I be there?
I will check, but most of the groups are already full. Maybe you joined very late, unless we shall just create more. Yeah.
Okay. Okay.
Okay, so let's go back to our work. Let's go back to our work. Yeah. So in summary, that has been. So you had to, although I've told you here, we don't shade like it is here. And this is what was supposed to be. Unless you have any question, we may end here unless you have any question. Teacher, why do we shade in biology? I said we do not shade in biology. We do not shade. And maybe if you are asking, why don't we shade? Now, shading kills most details. When you are examining a part, sometimes there are some lines within. So if you keep on shading, many details are lost. For example, just imagine if we had shaded the endodermis, okay? If we had shaded the endodermis here, in this case, here, if we shade the endodermis, that means the pericycle disappears. Let me play it for you here. And then I get a black pen. Now you see, this is the endodermis. I'm assuming I had drawn and then I had shaded it. If I don't you see that now it's like you have only drawn the endodermis and the pericycle disappears. Okay, so it doesn't make sense to shade. So it kills details. Miss Ghana has a question. When I ask you to unmute, yes.
I just want to ask you that, how can we know what a leaf is, a dicot or a monocot? You were explaining about it, but you gave us a question, draw a well-labeled diagram showing the transverse section of a bean root, but at that time, we don't know the picture of that root. We can't differentiate it, whether it is a monocot or dicot, without looking at the root. So what can we do?
Okay, thank you for that question, Ms. Ghana. Now, what you must know is in most cases, we use examples of plants which we know that everyone has seen, okay? For example, it is rare to find someone who has never seen a bean plant. You get it? So in school, you must have studied in practical, because it is there in the curriculum that when we are learning these things, you are supposed to look at the bean plant. So they know already that you have already seen how a bean plant looks like. And you know that even its classification that it falls under kingdom plant, phylum Angiospermatophyta, those are flowering plants, okay? And the class Dicotyledonea. So for you to classify it that way, you know features that make it placed in all those different groups, okay? So if you know that a bean plant falls under class Dicotyledonea, so it means, yes, a dicot, so it must have broad leaves, network veins in its leaves, like that. So even when you are drawing transverse section of its root, then its xylem is in form of a star shape. And those are some of the differences actually which they ask. In case they ask you to give differences between a transverse section of grass, any grass plant that you can think of, and a transverse section, or transverse section of a dicot. One of the differences is in the dicot, the xylem is in form of a star shape, okay? But in the monocot, they are just scattered, arranged in a ring. So you must know that already. Do you get it, Miss Ghana?
Yes, so teacher, you're saying that we have to study it. You have to study the roots.
Yes, it is. It is even there in the, we call it the syllabus. It is there that you must learn it.
Okay, teacher.
Okay. Yes, Agaba, you have a question? Agaba? Okay, Agaba has gone off, and I think there are no more questions. I think, is there any other? Yes, Tendo Brenda?
Can we draw using a pen?
No, according to the according to the guidelines of UNEB, as long as you are still following the UNEB, Uganda National Examination Board, and you're going to sit in UCE, we do not accept drawings of a pen. We only, and even it is always there in under the instructions that for writing, you are only supposed to use a blue or black pen, okay, for writing. If you use a red pen, they don't mark your work, they cross it out. And for drawings, you should always use pencil. Those are very clear instructions from UNEB.
Okay.
Okay, and then yes, Victoria.
Teacher, is it a must to put magnification below every diagram?
Yes, you should always do it. Because it gives a scale of how big or how small it is compared to the original which you had. So most of the practical work you should always include, and there's always a mark for actual magnification. So if you don't put it, you are losing that mark. Okay. Okay, I'm not seeing any more questions. And you can see it's already time. I've liked your questions. And I think goodbye and have a blessed day. Goodbye to you and everyone. Hey, Mulunji, you are saying bye or a question?
Teacher, some of us, we didn't get our marks.
Yes, you are going to get them. You are going to get them, please. You're going to get them. Okay. Chazoto, I think we can end the meeting, and yeah.